Hello, prospective graduate student. My name is George Woodward. You may have seen my recruitment video already, but now I'm going to give an overview of the functions and interactions of selenium protein K, or in other words, I'm going to give a quick overview of what I've been doing in the Rosowski group for the past year. This will range from software predictions I've done to actual research to future directions in the group. Um, the focus of the Rosowski group is what is known as selenoproteins. These in humans comprise 25 known proteins that all contain the amino acid selenocysteine. Um, Selenocysteine is very similar to cysteine, except instead of sulfur, it has a selenium atom instead. And this human selenoproteome comprises proteins that have functions ranging from antioxidant behavior to thyroid hormone regulation to calcium signaling to protein quality control. Um, and in protein quality control, you have seleno K and seleno S. These are two of the main foci of the Rosowski group, and seleno K, in my case, has been the focus of a lot of my research in the past year. Uh, this protein, seleno K, that is a small and unique protein, and it only contains 94 residues and is 11 kilodaltons in size. You can see I have spaced this out 10 residues at a time to make it easy to see. And you can see that U, which is the one letter code for selenocysteine, is at position 92 out of 94. It's predicted to have dual functionality. I'll get to that in a second. And it has a highly disordered region. You can see here from this figure in Polo et al. 2016 and from this prediction on a website called the elm.eu.org website, eukaryotic linear motifs, that about half of seleno K has a highly disordered region. This means that there's no discernible structure, that is no alpha helices or beta sheets that we can predict in this region. And this makes it very difficult to do structural studies of seleno K, and why figures like this one from Polo et al. Are exceedingly rare to find for the whole structure. In many cases, when you try to do predictions of seleno K online, such as with the Fire server, PHYRE, that is not fire with an F, you can only really get the helical domain right here. And this helix is actually amphipathic, meaning that it's half hydrophobic and half hydrophilic. Therefore, it's actually not transmembrane. It's not a transmembrane protein, but seleno K is actually a peripheral membrane protein. The first function of seleno K is calcium channel modulation, and this has been extensively studied by other groups. This seleno K associates with the endoplasmic reticulum or ER membrane and physically interacts with a protein called DHHC6. Now, this is what's called a palmitoyl transferase, and it slaps on a palmitoyl group by taking palmitic acid and puts it onto the calcium channel receptor right here. This opens the receptor and lets seleno K in turn modulate calcium flux together with DHHC6. This is very important for immune system control and actually the growth and inhibition of several cancers. This has been extensively studied by the Hoffman group, and it's a very well-known model, except right here, seleno K would really be better shown as a peripheral membrane protein on the bottom of the membrane right here, not just going right through. The other function of seleno K, and which is of much more interest to our group, is protein quality control. Specifically, it works in what's called the endoplasmic reticulum associated degradation of proteins pathway. It's a mouthful, so we abbreviate as ERAD. Misfolded proteins, in turn, are actually managed by the ER. And many proteins, in fact, are actually synthesized in the rough endoplasmic reticulum as opposed to the cytosol, that is, many membrane proteins. If a protein is misfolded in the lumen of the ER, it's targeted to the ERAD pathway or the ERAD complex. And this complex is comprised of Derlin-1, HRD3, YOS9, and of course seleno K. But you'd also see seleno K in this figure right here. So what's thought to happen is seleno K works to translocate the misfolded protein into the cytosol, where the misfolded proteins then ubiquitinated, um, represented by the UBs right here, and it goes to the proteasome for degradation. This is extremely important for making sure misfolded proteins don't accumulate and then eventually aggregate as plaques in your cells and cause deleterious health effects. Um, we know now that selenilyl K is involved in many protein-protein interactions and is predicted to interact with many more. Um, one, of course, is DHHC6, which modulates calcium flux. The other is Derlin-1, which is just one example of the proteins in the ERED complex. And another is 11433 protein. I haven't mentioned this before, but selenilyl K is predicted to interact with this. And this protein, the 1433 that is, binds to signaling proteins and thus is involved in several signaling pathways. And this, in our contemporary research, could be a good bridge to go to once we get past what I'm about to talk about on the next slide. Protein-protein interactions, or PPIs, don't always happen with just the proteins physically interacting. Sometimes the proteins need to be modified after they've been translated. And one of the main ways that proteins are modified is, of course, phosphorylation. Um, I've used a software called the GPS 5.0 system to predict where selenilyl K is actually phosphorylated or where it could be phosphorylated. I got many different results across the protein, and in fact, I got results for pretty much every serine or threonine or tyrosine residue. However, several in particular kept repeating, and these what I've shown in color are just some examples of the residues that kept coming up. And several of these predicted kinases are expected to work on selenilyl K as well. We actually have all these kinases as vectors in our lab. I've physically worked with DYRK and PKA so far, and as also I have done a little bit of work with MAP kinase. I found already that MAP kinase doesn't actually phosphorylate its own substrates unless MAP kinase itself is phosphorylated. And based on what happens in the MAP kinase signaling cascade, if you recall for cell biology, that actually makes sense because the cascade can't finish until the kinases themselves are actually phosphorylated. So one example of a protein interacting with phosphorylated partners is the 1433 protein, which has a phosphate binding pocket here. I've highlighted in cyan, consisting of a lysine, two arginines, and a tyrosine. Selenilyl K has two 1433 motifs. So we're represented in here. I've highlighted one of them. And again, I'm only able to use software to predict a small portion of the whole structure of selenilyl K. Again, because of its somewhat hydrophobic nature and very distorted nature, it's very tricky to determine what the full structure looks like. We can detect phosphorylation where we thought we could detect phosphorylation by two different ways. That is, one way would be to see if the electrophoretic mobility on one of these glycine or strengthening gels is actually affected by the protein being phosphorylated. In theory, you could see if that actually happens because the protein would then have a very large negative sphere of electron density just being slapped onto the sequence. But it turns out this is much trickier than we thought because at low pH, what we need to have our samples at when we terminate the phosphorylation reactions we came up with, you can't actually see what happens on the gel because low pH is incompatible with these gels. We could revisit these in the future by making the pH of our samples after terminating them very high, but a better way would be to just keep using a mass spectrometry. This is very easy to do, very quick and easy. You can very clearly see peaks of interest if you have a very pure protein. This signal to noise ratio could be a little better, but this is just one example of the many mass spectra I've been getting in these phosphorylation studies with selenilyl K. This in turn is actually not
Flannel K from a phosphorylation study. This is just after seeing if I had a strep2 tag cytosolic flannel K, and based on the math right here, that 6787, it looks like I did actually get it. So if you get a protein that's 100% phosphorylated, for example, at one site, you would predict that you could add 80 um, dolphins onto the mass of the protein. So for this example, you could just add 80 to 6787, and you could expect to see 6847. And you can see a slight shift in that case from here a little to the right. That doesn't always happen. You don't always have 100% phosphorylation, but that is a totally ideal, totally theoretical thing that will happen. Future directions to consider. That is why would phosphorylation be important to study? Why keep doing PPIs? Well, phosphorylation and in turn PPIs for Seleno K are important because we could actually see how the activity of Seleno K is affected and how other properties of it are affected. Like for example, maybe the not just activity but stability of Seleno K is affected. One example is PIN1, which is an isomerase that catalyzes the transition of proline into from trans into cis. Trans right here and cis right here. And this is just the PDB structure that I got of PIN1 for humans. Uh, it's thought that PIN1 could actually act on Spleno K because Spleno K has a serine um, proline motif, which is what PIN1 recognizes. And PIN1 only recognizes that motif if the substrate is phosphorylated. So a possible future direction could be if we can determine that specific motif is phosphorylated, PIN1 may actually act on that and in turn modulate K's activity in possibly the ULAT pathway. These are some of the references I used throughout my presentation here. Polo AL, Frederick and Hoffman for the DHHP6 pathway, and they picked a paper from which I used for a figure from the ERAD pathway. If you have any more questions, you can always look on our group website, and you're more than welcome to email any member of our Wazowski group, and you can email me as well.